Hi everyone, welcome to Biogram Academy. So here we are about to start our first chapter on genetics. Our first chapter is Principles of Inheritance and Variations. Alright, so what are we going to see in this chapter? This chapter obviously deals with the basics of genetics. Alright, so we are going to see about what are the rules of inheritance, what are the reasons that in spite of inheriting genes from our ancestors, still we have some variations. Why do we have those variations? And what are the exceptions to the laws of inheritance? What are the genetic disorders? Why do they occur? All right, so we are going to see all of these things in this chapter. So in this video, we are going to just see about the basic terminologies of genetics, the terminologies that will be used completely across this chapter. So we first of all need to have the idea of the basic terminologies of genetics, and then we can dig deeper into the rest of the topics. Okay, so let's get started. So the first terminology is heredity. What is meant by heredity? Heredity means transmission of characters from the parents to their offsprings or from one generation to another. Okay. So it is meant by the transmission of characters from parents to offsprings. Alright. So how does this transmission take place? This transmission takes place through genes, right? So genes are basically transmitting the characters from one generation to the another generation. The another important terminology in this chapter is variations. So if genes are getting transferred, if genes are transferring the characters from one generation to another generation, why do we have variations, and what do these variations mean? These variations mean they are differences in characteristics shown by individuals of a species and also by offsprings or siblings of the same parent. Alright, so it is the differences in characteristics shown by members of same species or by siblings of same parents. Alright, so these variations can be in the form of physiological variations, morphological variations, there can be cytological as well as behavioral variations as well. Alright, so the variations can be at different level. So the variations means they are differences in characters that are shown by members of same species or by siblings, that is children of the same parent. Alright, and the other very important definition of gene. Alright, so what is meant by gene? Gene is simply a fragment of DNA. It is a fragment of DNA that codes for a particular character. Alright, so gene means it is a fragment of DNA. A very simple and very precise definition. It is a fragment of DNA that codes for a particular character. Alright. Okay. So related to the terminology gene, we have another term which is known as chromosomes. Right. We often get confused between these related terms. What is meant by chromosomes? So chromosomes, from the word itself you can understand the the word chroma. This chroma means colored, and somes. This word comes from the word soma, means body. So chromosomes are basically some colored bodies, right? So physically they are some colored bodies. It means they have a tendency to absorb stain very quickly. That's why they are known as chromosomes, because that is the way they were detected. Because there was something in the nucleus that was catching the stain very quickly and efficiently. From there the word chromosomes was coined. But how can you define it in relation to genetics? How can you define it? Chromosomes are condensed structures of chromatin threads. Alright, a very simple definition is chromosomes are condensed structure of chromatin thread. Now what is chromatin thread? So the terminology goes on. Now what is meant by chromatin thread? Chromatin thread is nothing but DNA on its way to become the chromosome. Chromatin thread is the transient state, chromatin thread is the term that we give to the transient state when DNA is getting winded and getting coiled to ultimately form the chromosome. Alright, so that is the chromatin thread. Alright, so the next one is, what is DNA? These are very simple terms, but we need to just have a small brush up before starting the chapter. DNA by definition is 2' deoxyribonucleic acid, right? So it is 2' deoxyribonucleic acid. So here comes the word DNA, right? So DNA is deoxyribonucleic acid, fine. And apart from that, what is it actually doing in our cell? It is a polynucleotide structure, right? It's a polynucleotide structure that is double-stranded, that is helically coiled and contains all the genetic information. Okay? So DNA is a polynucleotide structure that is helically coiled and double-stranded and contains all the genetic information. Alright, so that is meant by DNA. So DNA is a polynucleotide structure. Why do we call it polynucleotide? Because it is made up of polymers of nucleotides. Nucleotides come together to form a polymer, and that is a DNA. It is a double-stranded structure. It is helically coiled and it contains all the genetic information. So we are going to see in this chapter how is DNA able to contain the information, in what form does it contain the information, and how the information is deciphered in our body. How are the information getting finally expressed? We are going to see about all these details in this chapter. Alright. So the next important term is genome. What is meant by the word genome? Alright. So by definition, genome means a haploid set of chromosome. A haploid set of chromosome. A very simple definition is haploid set of chromosomes in a gamete is called a genome. Okay. So this can be a definition. There can be another definition also. That is a complete set of genes or genetic material present in a cell of an organism is also called a genome. Okay. A complete set of genes present in an organism is also called genome. Both the definitions mean the same. A haploid set of chromosome or complete set of genes. Now what does that mean? Let us see that. why are we using this terminology haploid set of chromosomes? See, I can explain it. We have 23 pairs of chromosome, right? A human cell has 23 pairs of chromosome. So when we are saying the word pair, what do we mean by that? Pair means for every chromosome there is another replicate set, right? There is duplicate set of every chromosome. So whatever information is present in one of the chromosomes is also present in another, is also having another replica of the same, right? We have two replicas of each chromosome in our cell. So what if we take only one of the replicas of all the chromosomes, all the 23 pairs? Out of the 23 pairs, you take out one chromosome out of each pair. So you are going to get all the information present in the 23 pairs of chromosomes in just that one set, right? When you are talking about 23 pairs, that means you are talking about the entire diploid set of chromosome. But if you just take out the haploid set of chromosome from here, if you just take out the haploid set, you will get all the information present in the individual. Because the information that is present in the haploid set, just another set is also present in the diploid set. Is it? Isn't it? So just the haploid set of chromosome will going to give you all the information of all the genes of that organism. So a haploid set of chromosome in a gamete is meant by its genome, because this haploid set is going to have all the information, and that is what you can call as genome. And the same thing you can also write it in an elaborate way. A complete set of gene present in an organism is also called genome. Alright. So I think I am clear about it. So from here, another important term that we come across is alleles. What is meant by allele? Yeah. Just now we talked about haploid set of chromosome. Why? Because whatever information that is there in our cell, we are having it in duplicates, right? In any sexually reproducing organism, in any diploid cell, we have all the information, all the genetic information in two sets, right? So just like that, for every gene also we have two sets of the same gene, right? Why do diploid organisms have every information, every gene, every information in two sets? Because one set is obtained from either of the parents, right? So that means for every gene that we have in our body, for every gene also there should be two pairs of the same, there should be two copies of the same gene, right? So from here we come to the word allele. Allele means alternating forms of the same gene. So alternating forms of a gene are called alleles. Alright. I can just make all this small terminology easier for you. Suppose we are talking about gene A. So gene A is obtained by an organism from both of its parents, right? So suppose both of its parents have given one allele of gene A to the offspring via the gametes. So suppose the two alternating forms are small a and capital A. Alright, both should be A. So one is small a, another is capital A. So that means this offspring, offspring X has gene A in the form of capital A, small a. Alright, that means offspring X has the gene A in the form of capital A, small a. So this thing, capital A, small a, is going to represent gene A for this organism. Am I able to understand, able to explain you? So from here we come to another important things, that why are we writing one as capital and another as small? We usually denote the dominant form of the gene, dominant form of the allele with capital letter. So that means capital A is the dominant allele and the smaller letter is the recessive allele. Alright. So gene A can be represented as capital A small a, where capital A denotes the dominant allele, small a denotes the recessive allele. Alright. So from here you again come into some new terminologies, that is genotype. What is meant by genotype? Genotype means the genetic composition of a gene. Alright. Genotype means the genetic composition of a gene. So genetic composition of gene A. The genetic composition of gene A is here capital A small a. Okay. And from here another similar term is phenotype. What is meant by phenotype? Phenotype is expression of the gene. How the gene will be finally expressed, that will be the phenotype. The expression. The expression of a gene is phenotype. Alright. So I can explain you genotype, phenotype with another example. Suppose we are talking about the trait height. Okay. We are talking about the trait height of an organism. Okay. Suppose height is represented by gene T. Height is represented by the gene T. So gene T might be again having have capital T and small t as two alleles, right? So if gene T has whatever combination, say let us talk about all the possible combinations that gene T can have. Gene T can be present in capital T form. It can be present in capital T small t. It can also be present as small t form, right? So gene T, that means, can have 3 genotypes. Now you get to know the term genotype. Gene T can be present in 3 genotypes. Either it can be capital T, capital T small t, or small t. So how can these be expressed? What will be the expression of these genotypes? How will be the expressions different of all of them? Let us talk about it. We know that capital T is the dominant allele. Whenever we are representing a dominant allele, we use capital word, right? Capital letter. So capital T should denote tallness. Okay, it should denote tallness because capital T is dominant and dominant trait in height is obviously tall. Similarly, small t is not tall, that means it is dwarf. And what about capital T small t? So capital T small t, it is having a mixed combination. This genotype is having a mixed combination. But since we know that the capital T is dominant over the small t, so that means capital T small t should again be tall, right? Because capital T is the dominant allele. The dominant allele is expressed and the recessive allele is not expressed. So that's why we are again going to get tall in case of capital T small t combination. Okay. So from here you can understand that these are the genotypes and these are the phenotypes. Okay. These are the genotypes we are talking about and these are the phenotypes we are talking about. This is the difference between a genotype and phenotype. Now again, to make you more clearer, how do we address these genetic combinations? Capital T, since this genotype is having both the alleles same, it is having both the alleles same and both the alleles are dominant. We use the word homozygous dominant. Homozygous dominant for this combination. Alright. For the second combination, capital T small t, because it is not same. The two alleles are not same. The two alleles are different. That's why we use another terminology. We won't be using homo for this combination. We will be using the term hetero, right? So it is a heterozygous combination. It is neither dominant, it is neither recessive. So only heterozygous genotype. Okay. And the last one is again homozygous. Again both the alleles are same. That's why we will be using the term homo. It will be homozygous, but it will be homozygous recessive. Right? It is homozygous recessive. Alright. So these three are the genotype and these three are the phenotype. So these were the basic terminologies that we needed to be discussed before starting this chapter. So in our next video, we are going to dig deeper into the details of the other topics. So in our next video, we will be dealing with Mendelism. So stay tuned to my channel and please subscribe. Thank you.